even listen to podcasts while being physically active, etc. So here I will really, first of all, showcase you data which are not published, and some are published, because otherwise it's not a story. So I will talk about evidence-based. That's very important for me. Geromedicine or health longevity medicine, I think we still have to decide how we term ourselves. And I will talk about especially interventions in the second part. I have a couple of disclosures. I have not only affiliations with bigger companies, but also have G Longevity, which is a private longevity clinic, and for the ones we are hiring. And I'm the Chief Medical Officer of NU. Most importantly, and that's most dear to my heart, is the HLMS, Healthy Longevity Medicine Society. Yesterday we had our General Assembly. Most of you were there. Thank you so much. For the ones who were not, this is your chance. Now the iPhone's out, and scan it, become a member if you're actively involved in the healthy longevity medicine field. What we did 1.5 years ago, and it's now in submitted that paper, we defined what is healthy longevity medicine. And that's most important. And here you see the definition. Healthy longevity medicine is optimizing health and health span by targeting aging processes across the lifespan. So that's important. So we need to optimize health span. We need a good definition for it. And we have to understand the aging process, which means that whatever is happening in the main room, we need to translate. So we shouldn't have two tracks at this moment in time, because you are missing out the biology of aging, because we as clinicians have to understand the biology of aging to then translate that into human trials, whatever it's NS1 or randomized control trials, to then implement into clinical practice. So that's very important. And very important is also that we should do it across the lifespan. We had many discussions when is the right referral time to come to a health longevity medicine clinic. I think nobody knows. I would say during the life course, because everybody is aging all the time, because cells are dividing. So really the scope of HLMS is to build standards, etc. So you all know, look at the website, it's all available. And I will now talk about the first working group we have out of HLMS. We met 2.5 years ago. I think it was a meeting out of desperation. We all wanted to set up healthy longevity medicine clinics in publicly funded hospitals. And what we did was a couple of people from the NUS site and Sheba and also the Mayo Clinic, we built a framework. How do we actually build publicly funded hospital systems? What does it involve? And it's published, and Geroscience Sarah is the first author, so please have a look. What we also did is not only defining what should the workforce look like, but what is the trajectory within a health longevity medicine clinic. And very importantly, and that's also dear to my heart, lots of dear to my hearts, is that we are diagnosing something, we have good investigations, then we have treatments, and then we are assessing again. We are looking at what the outcome is of our interventions. And that's most important, and that's very often missing at the moment, in my view, in health longevity medicine clinics. What we do, so this is also published, what we do in Singapore, which is not published, while having quite resistance, I would say, from the conventional healthcare system, having a publicly funded hospital clinic, how do we involve and how do we merge that with conventional healthcare systems? So now we are thinking about how do we involve it into primary care? When do we actually have to refer to a health longevity medicine clinician? Or when do we have to scale up what GPs cannot do? So I think we at the moment in Singapore at the time is, what is the role of health longevity medicine? What is the role of GPs? How can we merge the efforts? And how can we actually not duplicate everything? And I think that's most important, because in the end, everybody is aging and it should be available, the care for everybody, and has to be cost effective. And that led to a question I got 2 years ago when I had to defend a business case, and they asked me, Andrea, who is coming to a health longevity medicine clinic? My answer at that moment in time was, the ones who are interested in their health. And I thought, oh, Jesus, this is really not good enough. Like, I am an academic person. Like, I have to throw with numbers, and I have to do something. So we started the HALO Consortium, the Healthy Longevity Understanding Consortium. We have beautiful people working with us around the globe. If you want to join, let us know. And what we really want to achieve is 3 things. We want to understand the public awareness about health longevity medicine. We want to understand not only the perspectives, but especially the knowledge. Do they really know what it is? And we want to understand the barriers. And we created, and it took us 1.5 years, and now validated questionnaires to understand the 3 aspects with regards to health span, lifespan, and and knowledge around health and longevity medicine. So I will only present the data from Singapore because that's new. It was released 10 days ago, so please excuse that not all the tables are really nicely done. What we did is we looked at Singapore and we wanted to have a snapshot of 2,000 individuals living in Singapore being most unbiased. And how can you do that? You're going to somebody's home, you're knocking on their door, and you say, please, you really have to participate. So what we did, we put Singapore into different districts. We looked at socioeconomic classes, we looked at religions, we looked at ethnicities, and then we mapped it out where to knock on the door. And that's what our team did. So you really see that we had 20 volunteers knocking on the doors and applying the research. I will only showcase you the data with regards to what is the knowledge and percentage about lifespan, health span, do people know what we are talking about, and would you come to a health longevity medicine clinic? Next to that, because we have a big consortium, we also wanted to know, is it really necessary having lots of manpower knocking on the doors? Can we also do it with surveys? So that's the reason why we have 1,000 online responses too. So here are the first results, never been shown. The online survey first. So 1,000 individuals, we asked them, do you know what lifespan is? And the majority said, yes, I know. So that's the first bar, 88%. Then we asked, do you know what health span is? And roughly 40% knew, yes, I know what health span is, which is great. And now, of course, the excitement, we ask, okay, would you like, and we gave a little bit of information what health longevity medicine is, would you like to come to a clinic? And even if they would pay for it, it's 2/3 of the population nearly. So that's, I think, whoa, I didn't expect that. I thought maybe 20%, 25%. If we are now, of course, looking at the door-to-door, a little bit different, but absolutely the same numbers or the same trends. 80%, 40%, and more than half knocking on the door, independent of a socioeconomic class, would love to come to a health longevity medicine clinic. Guys, we have a problem because we are not prepared for it. So people want to know what it is, want to come, they are eager, and now we have to deliver. And I think that's part of HLMS. And of course, we would love to know who are the people who said yes versus no. These are the first analysis. Who is interested in healthy longevity medicine? And I would say nearly everybody. We don't see so many predictors. It's a little bit more, if you are more highly educated, if you have the housing, if you have more rooms, it's a little bit a status of socioeconomic class. They would love to come a little bit more. But if you're looking at it, and this is a population of a middle-aged population, so really general population of Singapore, 45 years of age, all want to come. So I think that's nice. What do we have to deliver that we really can change healthcare? So we need diagnostics and we need interventions, of course. So what we did in the past, I would say months, with a couple of people is really thinking about how can we deliver care that in the end it could be implementable. And we also looked at especially the genetic contribution to healthy longevity medicine. And we termed gerogenes and gerosuppressors. And that was a little bit after a visit especially to the Middle East, where there are lots of projects where everybody is being genotyped. And I got the question, what do we do with the information? And how can we apply that to health longevity medicine? So that's summarized in a paper which came out a couple of weeks ago. Please have a read, where we really try to map out how can we link the genomics to the clinical biomarkers of aging, and in the end, even to interventions. Have a look. In the end, the WHO is our best friend and should be our best friend. And I think we have to be much more rigorous in understanding the language of the WHO and ICD codes, so the International Classification of Diseases. And that's the reason it's also published that I think we have to use the words intrinsic capacity and vitality capacity much, much more, because these are the terms already embedded into the codes, which means that if we are now transferring these codes into language we do use in clinical practice, we could even use the codes. And these codes came out of a long history of defining frailty, which was not successful. And then intrinsic capacity, vitality capacity came here. And I think it's beautiful. There are now working groups really looking at age-related ICD codes with the Cardiff University. If you are interested to join these kind of working groups, please let me know. Most importantly, what I'm struggling with in clinical practice is what to measure, because protocols are lacking. So this is really a problem. And I just give you one example. I know that many of us here in the room are working on it. We very often forget how to measure female reproductive aging, or how to measure oral health or microbiome health or whatever. We don't have the protocols. So within HLMS, we should go together and come together and write these protocols. And these are just examples. Very important also, if you're doing randomized controlled trials, I think we have to agree on the protocols, because otherwise we are just comparing apples with bananas. And that's not good. So let's come together, share the protocols. It will not harm us, but it will excel us to really be able to go into clinical trial networks which are worldwide. That's the reason why we now established a relationship, a collaboration with IQVIA, which is very often used in randomized controlled trials in oncology, in other disease subsets. Also very important is that we really do not know how to measure things digitally. Everybody here has devices. I see lots of smartphones, make photos, it's very good, but how do we use it for health? So Jessica, who's also here in the room, I think, wrote a guideline how to choose devices and how to use them. So please reach out to her how to do it. That's published. I think it's hugely important that we know what we measure and how to do it. Sorry, these were lots of questions. Now we are going to the future, and I will showcase you in my last minutes a couple of randomized controlled trials, what we are doing at this moment in time. The first one I'm very excited about is our heart study, where we are testing SGLT2 inhibitors in healthy individuals to lower not only the HbA1c, but especially improve, hopefully, the VO2 max. We are randomizing, of course, and we are including quite healthy people to really, with the understanding, can we even optimize them to have better outcomes, hopefully in 30 years' time. That's not our primary outcome, but it's just a very, very short study. Then we have our alpha-ketoglutarate work. It's just one of the supplements we are testing. We also have, in terms of repurposed drugs, we are testing rapamycin, but I just give you snapshots what we are doing. I think in the past 2 years, we really understood how to do clinical trial designs, how to include individuals. Alpha-ketoglutarate is a nice molecule. I think we have to really test if it really works, and that's the reason why we are investing hugely, not only giving it to middle-aged healthy individuals, but also now we recently started a RACT in individuals perioperatively. They are coming and undergoing a heart surgery. We give it beforehand, a sort of prehab, but then with supplements, and see if the outcomes are better. We are hugely working on nicotinamide mononucleotide. There are lots of systematic reviews at the moment going. This is all data where we already showed that NMN supplementation it was a very, very, very small study, that it could lead towards better functional outcomes and quality of life in middle-aged individuals. We use this data to really get a better understanding about the physiology of NAD of NED, how much actually change we need to have clinically relevant outcome parameters. Also, that is published, where we really show that you should at least see an increase of NED levels by 15 to 20 nanomole per liter. Because then you really, we were able to predict who is responding and non-responding. This will lead to the CURATE study. Yesterday or the day before, we heard about NS1 study, so RCTs is one, but can we do other trial designs? So we are now at NUS really embarking on different trial designs where we're including one individual, giving the supplements a washout period, giving it again, washout period, giving it again. And we are doing it with the NS1 Institute with Dean Ho, which is, I think, super exciting. We are starting with Vera, very exciting study to really manipulate THIRD6 with seaweed. This study is now being submitted to ethics. Middle-aged individuals, also a little bit pre-frail, sorry, it's not really middle-aged. I think also 50 to 80 is middle-aged if you're becoming 120. Pre-frail individuals, also here randomized controlled, because we do not know if the NS1 approach is really the one to head for. So please let me or Vera know if you have any questions. The last one we are embarking on, it's with ethics at the moment, is multivitamin and mineral use. There's lots of discussion at this moment in time with regards of is it good or not good to use multivitamin and minerals because it's associated with not much mortality benefit or survival benefit, but maybe even mortality. So Daniel, who is also, I think, here in the room, wrote an umbrella review what the potential benefits are of multivitamins and minerals. You see them here. It's quite unknown, I would say. We are embarking on really optimizing micronutrient levels in individuals who are middle-aged and healthy, really defining, and that's a more an academic exercise, what is normal and what is abnormal, that's quite clear, but what is optimal for you, based on your lifestyle, based on your needs, that's what we are doing at the moment in the CEDERA study, together with Halion, in testing if a multivitamin and mineral pill can actually decrease the epigenetic age of middle-aged individuals. Last but not least, very importantly, this year in February, we had a fantastic conference. There is a follow-up in February, together with the Founders Forum and Longevity Technology. We will have the next iteration of our conference. Please come. There are also some flyers. We have intensive courses for healthcare professionals in the same week, and we have an open day, and it will just be, I would say, fun, which is great, as the ARDD at this moment in time. So I would say the summary, let's be clever, let's take some risks, let's have fun together, and let's work together, because then we can really shape the future of our beautiful body. Thank you.
Thank you so much. Thank you, Andrea. Thank you very much. We can take one or two questions. Anybody?
What is the standard protocol? We'll have the lifestyle. It's kind of ethical. We have to recommend it. There has to be some kind of base, so that's not a confounding factor.
So what is the protocol? I would say not to do, but to measure lifestyle. So we have digital phenotypic characterizations, all our studies. So speak to Jessica, she is sitting there. So all our participants are equipped with an Aura or with a Fitbit, etc. So we are watching them. And if you're doing randomized controlled trials, it should not matter because you are randomizing it. In the NS1 approaches, it's very, very important. So there we are going to measure very, very heavily.
Great. Quick one.
Yeah. Thank you so much. In pharmacological trial, usually there is a pre-trial where one tries different doses, different timing, different populations, but here I can see all these factors are aggregated together. So how you design these trials?
Yes, that's a good question. So what do you need in terms of, do you need a phase 1 and 2A and 2B study, etc.? We do that. I just have no time. So we also do phase 1 studies. We are testing NED changes. So we are doing all of this. So yes, you should know what the dosage is, the frequency, etc., and then you should go to a bigger trial. But these are all not phase 3 trials. So these are all phase 2B trials. I think we need the consortium here. Please, if you really would love to build such a trial network and be part of it, let me know, because that's what we need. And then we can go to a phase 3 and eventually with lots of clinical partners into a phase 4 trial. That's what we need. But we also do the phase 1.
Great.
And Evelyn, thank you so much for giving me the chance to speak today. We've been working on a very interesting project with Evelyn. It's the use of epigenetic clocks in clinics. So today we can measure blood, the liver, the heart, lungs, everything, and we can give each one of them an age, a biological age. And it's wonderful the work that's been going on so far. It started in 2011 with Blockland creating the first epigenetic aging clock, but so far, there has been lots and lots of research, but little application of these clocks in the clinic, which is what we would want to emphasize today. So the reason why clinics would benefit so much from these clocks is that they not only give the biological age, but they can also tell the pace of aging, so that people can track how well treatments are working, and therefore lay out treatment trajectories for their patients. Now, what we did is that we collected, through all existing literature, all of the epigenetic aging clocks that currently exist. And epigenetic aging clocks, they differ from other biological aging clocks. These are the most well-studied clocks. They have a lot of evidence for them. They're based on CpG sites, which show the expression of certain genes, and you can measure this expression and compare it between different people. And what we did was we analyzed and really dug deep into each one of these clocks in order to understand which one is the best, which one has good characteristics, and which one is useful for an actual clinical setting. And reviewed them according to strict criteria, and from all this research determined what the criteria of a perfect, a dream epigenetic aging clock would be. So here's a short diagram, small diagram of what we've been looking at, and it shows you the most fundamental epigenetic aging clocks currently in the field and what's been going on. So you can see a lot. The ones that are the earliest have the most simple features, but they lay really the base and the foundation, whereas the later ones are based on deep learning and artificial intelligence and many more complex algorithms. And this is the criteria by which we analyzed each of these clocks. So we had to make sure that the method of making these clocks is reliable. We looked at the number of CpG sites that each clock used, and we also considered, for example, the sample number, how much data did people actually have when making these clocks. We ensured that there were different tissues studied, for example, blood, saliva, and we also looked at the applicability to different cell types, so whether people in the clinic will be able to use it just for one tissue type or not, or others. Of course, it's important to make sure that these clocks are good for males and females, and as well for different ethnic origins too. And we also evaluated whether these clocks were looking at disease-related effects on aging, among other factors. From there, we devised what the dream epigenetic aging clock would be. And these are the different criteria that we came up with. First of all, it has to be easy to implement and easy to use in the clinical setting. Otherwise, it won't be accepted as well. It has to require a minimal number of samples in order to actually function, but with high accuracy. What's important is that it predicts mortality. So how much a person, for example, has left to live based on this clock. This is important. It's one of the most fundamental measures for aging. Of course, it has to evaluate things like psychological age. That's related to brain aging. And we want the results to be delivered rapidly. We determined that there is a trade-off between 3 different factors. So first of all, the accuracy. So how well can we actually, how proper this clock is and how accurate its results are, how much can we believe it. Then there is feasibility. How easily can we actually make this clock and implement it? And finally, predictability. So can the clock predict things like mortality? And the next steps are now to evaluate non-epigenetic aging clocks. There are so many different ones that we can look at and consider for the clinic. And finally, designing this dream clock. Thank you.
Thank you so much. Thank you, Andrea. Great work all the year long, despite being a Caltech student. It's just pretty amazing. Thank you so much.